Hi, my name is Muhammad Iskandar bin Montafandi. So today I will give the introduction for our topic is the titration of drug in pharmaceutical industry. So let's go, guys. So the introduction is the titration is a fundamental analytical technique employed extensively in the pharmaceutical industry for the quantitative determination of drug concentration. This precise method involves the gradual addition of a titrant, a solution of non-concentration, to a solution containing the drug until the reaction reaches a known endpoint, which can be detected visually or by using instrumentation. And the endpoint signifies the completion of the reaction, allowing for the calculation of the drug concentration. So, in the context of the pharmaceutical, titration is crucial for ensuring the accuracy and consistency of the drug formulation, and it is used in various stages of drug development and manufacturing, from assessing the purity of the raw material to confirming the potency and stability of the final product. By providing reliable and reproducible results, titration helps maintain the high standard required for pharmaceutical products and ensuring their safety and efficacy for consumer use. So that's all from me. Thank you.
In theory, titration in the pharmaceutical field, especially in medicine, allows us to accurately measure the concentration of active ingredients in a medicine. So this allows the pharmacy department to adjust the dosage precisely. Furthermore, titration ensures safety and efficacy of the medicine itself. By giving precise adjustment in dosage, we ensure that the patient gets only what they need and nothing more. This also helps with quality control of a company's product. Through titration, we can get to know how much what the composition of the medicine, of the drugs, therefore making the quality consistent through every batch of production. And lastly, by doing titration, we can ensure the potency, composition, and purity of the product, and it can also help us get rid of all the unwanted elements in the medicine itself.
Some example of titration of drugs in pharmaceutical industry. The first one is dissolution testing. Dissolution testing quantifies the amount and weight at which a dosage form, such as a pill, capsule, ointment, etc., forms a solution. A drug's capacity to dissolve is crucial to both its bioavailability and therapeutic efficacy. Dissolution testing is done to assess how well drug ingredients are released from solid dosage forms. The concentration of the medication released from the dosage form over time can be determined using titration. This data aids in evaluating the drug's features of solubility and accessibility. The second example is acid-base neutralization. The term acid-base titration refers to a titration reaction between an acid and a base in which the concentration of one is found by fully neutralizing it with the other with a known concentration. In order to achieve the appropriate pH levels, acidic or basic components included in pharmaceutical formulations are frequently neutralized. To precisely determine the volume of titrant required to neutralize the sample and reach the desired pH, acid-base titration is utilized. The last example is impurity determination. A detected impurity content can be computed using the main component's peak area or by combining with a calibration curve or reference material. Pharmaceutical samples can have impurities identified and measured via titration. Degradation products, leftover solvents, or unwanted byproducts are examples of impurities. The concentration of the impurity can be determined by selectively reacting it with a particular titrant, guarantee a compliance to regulatory limitations.
Now in methodology, I will explain about dissolution testing. So first thing first, they will prepare the dissolution medium. So to prepare the dissolution medium, they need to prepare it according to the pharmacopoeia standard or as per the specific requirement of the drug being the drug that need to be tested. We need to prepare it according to the pharmacopoeia standard to ensure that the medium maintain appropriate temperature and pH throughout the test. Next, they will start to do calibration of titration equipment. Why they need to do this? Because they want to get an accurate result. So they need to do the calibration of titration equipment. Next, they will do the sample preparation. So they will weigh an appropriate amount of pharmaceutical tablets containing the active ingredients. And if necessary, they will crush or grind it to ensure the uniformity. Next, they will do the dissolution testing. So they will place the tablet in dissolution apparatus containing the dissolution medium, and they need to maintain the temperature agitation and other parameters as per the test requirement. At predetermined time interval, withdraw aliquot of the dissolution medium. Next, they will do the titration. They will titrate the withdrawn sample using the standardized titrant solution and appropriate indicator. And they will record the volume of titrant solution required after they reach the endpoint. Next, they will do the calculation. They will calculate the concentration of the dissolved active pharmaceutical ingredient in each sample using the volume of titrant solution and its own concentration. Last but not least, the data analysis. They will plot the dissolution profile of the pharmaceutical tablets over time and analyze the data to determine the dissolution rate, dissolution efficiency, and other relevant parameters. That's all for methodology.
In conclusion, drug titration experiments involve determining the exact concentration of the drug in the sample by carefully adding a solution of known concentration until the reaction is complete. It is possible to achieve precise and reliable measurements by carefully carrying out titration experiment. Accurate and exact readings can be achieved also, which yield to an important data for formulation and regulatory compliance. Pharmaceutical products are safe and effective because the titration procedure makes it possible to identify the active chemicals, availability, purity, and determine potency.